Good morning, everyone. It is my pleasure to introduce Professor Thomas Südhof for this morning's plenary lecture. Tom is a professor at Stanford University School of Medicine and an investigator at the Howard Hughes Medical Institute. When Tom started his laboratory in 1986, he was interested in studying how neurons in the brain communicate with each other at the synapses, and specifically the mechanisms that govern the precise release of neurotransmitters at the synapse. For this work, Professor Südhof received two highly prestigious scientific awards in 2013, the Albert Lasker Basic Medical Research Award and the Nobel Prize in Physiology or Medicine. More recently, Tom's research emphasis has shifted to understand how synapses and neural circuits in the brain are formed and how their properties are shaped, resulting in changes in various neurological diseases. Today, he will tell us about drug development for neural diseases. And as a fellow neuroscientist, I'm very, very excited to hear what Tom has to tell us. So a very warm welcome to you, Professor Südhof.
Thank you. I hope everybody can hear me, and I would like to express my appreciation for this very nice introduction. And I would like to express my apologies in that I have somewhat changed the content of my discussion with you here today, because it is so hard to do these things virtually. I thought it might be better to actually talk about a scientific project and to tell you a bit about our approaches to solve that project. And I thought that this would be particularly useful for younger scientists, because what I'm going to tell you illustrates our methods, our ways of trying to answer specific problems that are relevant for translational neuroscience, but address a fundamental issue. Now, another thing with virtual talks is that it's always more difficult to judge how long one is talking, because when you are talking to real people that were there instead of a computer screen, you actually get a feeling for time much better than when you're sitting alone in my office here and talking to electronic devices. So I hope I will finish my talk in time, but I may have to stop at some point because I'm running over time. So my lab is engaged in a broad effort to uncover the biological consequences of genetic changes that predispose to neuropsychiatric and neurodegenerative disorders, many of which are based on synaptic dysfunctions, which is really what we focus on. And today I would like to illustrate this effort by describing two representative projects. And I apologize, as I said already, for changing the content of this talk. So what I'm going to talk about in these projects are two very different molecules, genes, that are both involved in synapses and that are both predisposing to neuropsychiatric disorders. Let me talk about Neuroxin 1 first. So what are Neuroxins? What is Neuroxin 1? Neuroxins are presynaptic adhesion molecules that come in two major flavors, neuroxins, alpha neuroxins and beta neuroxins. There's three genes with independent promoters for both of these variants, and there's thousands of mutations in Neuroxin 1 that have been identified in patients with diverse neuropsychiatric disorders, autism, Tourette, schizophrenia. When you look at these disorders in general, all of these disorders are genetic to some extent at least. And there's been enormous progress in understanding the genetic landscape of these disorders in recent years. What you see here is a figure taken from a paper from the Daly Lab from medRxiv that illustrates the genetic landscape of schizophrenia, where you see the extent of an impact, the odds ratio in the y-axis and the allele frequency in the x-axis. And what you can see here, first of all, is true for all neuropsychiatric disorders, namely that there's a lot of different genes that have different degrees of impact and mostly rather uncommon in terms of mutations. The problem is that for most of these genes, we actually don't know what they do. Even for genes that have been studied for years, such as, for example, TRIO, which is a scaffolding gene, okay? Or let's say GRIA3, which is an AMPA receptors. Yeah, it's an AMPA receptor, but what exactly is unclear. What you can also see here is that the Neuroexin 1 gene is actually one of the more common genes. It's fairly, compared to others, it's fairly on the right, and it has a very high impact in schizophrenia. So this gene has a major effect on the risk for schizophrenia as well as other diseases. And when you zoom in on the mutations that happen, it occurs that the Neuroexin 1 mutations are almost exclusively deletions. And they're deletions of DNA in a large gene, the Neuroexin 1 gene. And as a result, they're always loss of function. And they're always heterozygous, or almost always. They're predisposed to many different neuropsychiatric diseases, and because they do predispose to many neuropsychiatric diseases, the question arises, how is that? As it happens, there's no correlation between the type of mutation, the deletion, the size of the deletion, the location of the deletion within the gene, and the disease. So what effects does this mutation have that it predisposes to disease, and why does it predispose to so many different diseases? So to address this question, we took an approach that we developed over the years in my lab in collaboration with other labs at Stanford, especially Marius Wernick's lab, and in which we use ES cells, embryonic stem cells, or iPS cells, and we make human neurons from them. And we study these human neurons as a function of mutations. We introduced a conditional mutation in the human neurons for Neuroexin 1 that mimics the mutations seen in patients. When we analyzed these neurons, we found, as you see here on top, that the neurons looked perfectly normal morphologically. They had normal numbers of synapses, normal dendrites, axons, everything. But not surprisingly, that expression of Neuroexin 1 was decreased by about 40%. After all, they had a heterozygous loss of function mutation. We then looked at functional effects, synaptic transmission, and what we found, as illustrated here, was a 40% decrease in synaptic strength, suggesting that there's a direct consequence in the strength of synapses as a result of the deletion of one of the two alleles of Neuroexin 1. So this is a discrete phenotype that likely selects an impairment of a specific step in synaptic transmission, and that may be amenable at some point to pharmacological intervention. We wondered what might be the molecular correlate, if there was any major changes in gene expression or in proteins. We found no changes, major changes in gene expression, tons and tons of minor changes, whatever they mean. But we did find one major change in proteins, which is an increase in the levels of this protein CASK, which is a scaffolding protein that binds to Neuroexin 1. The mRNA levels were normal, so this is a secondary event. CASK is a presynaptic chem kinase MAGO-type scaffolding protein that binds to neurexins of unknown function. This type may account for the change in synaptic strength. The robust and selective functional and synaptic impairment of the heterozygous Neuroexin 1 mutant neurons was unexpected. We didn't think that just losing one allele would get to such a major change. So we wondered if this change is actually detected in patient-derived neurons, not just engineered neurons. And to address this, we initiated a large-scale project where we investigated neurons that are derived from patients. In this project, as outlined here in A, we took the iPS cells from schizophrenia patients and matched controls that are only matched very superficially. They're genetically quite different. We generated these iPS cells. We then produce neurons from these iPS cells, and we use these neurons to study, as shown here, synapses as well as other neuronal properties. And all these experiments were done in parallel in 2 different institutions with different labs to ensure reproducibility, because, as you probably know, we have a reproducibility crisis in science. In parallel, we analyzed mouse neurons to determine whether the phenotypes that we saw in humans would be translatable to mice in order to enable efficient use of mouse as a model system. Let me first tell you about this COS platform, COS lab validation. So we do the patient-generated iPS cells exhibit the same selective synaptic impairment. I'm not going to go through this slide in detail. It would be too much time, too much work. I just want to focus on the key facts, which is that when we look at the reproducibility as analyzed in 2 different labs on different coasts, the West Coast and the East Coast, Rutgers on the East Coast, Stanford on the West Coast, we reproduced exactly the phenotype that we saw previously in these patient-derived neurons with independent lines, suggesting that indeed this phenotype is specifically observed as a function of the heterozygous mutation of Neuroxin 1. This large decrease in release probability just causes a major loss in synaptic strength that in turn is a plausible explanation for why these patients are predisposed to schizophrenia and Tourette and autism, depending possibly on other genetic mutations or accidental historical events in these people's lives. Is this also associated with a change in CASK protein levels? Yes, it is. What you see here is that we see the same change in the patient-derived neurons as we did in the engineered neurons. In addition, we tested several proteins we hadn't tested before, including a protein on the right called KIAT3, which is involved in kinurenic acid metabolism and which has been implicated in schizophrenia. And that you can see that this protein is also increased in levels, suggesting a potential pathway. So what emerges from all these patient studies is that we have a robust phenotype induced by heterozygous Neuroxin 1 mutations at the very fundamental level. This is much stronger than what we had observed in primary mouse cultures. So we wondered if this was really something that's human-specific. And this is particularly important because it's relevant for using mice as models. And so what we did in the second part of this project, as shown here in B, is that we analyzed in parallel identically generated mouse and human neurons with the identical mutation to test whether they would have the same phenotype. And in a nutshell, what we found is shown here in this direct comparison, left are the humans, right is the mouse, that in the human cells, the phenotype was reproducibly there between different engineered mutations within patient-derived neurons, whereas in the mouse, there was no phenotype in the spontaneous MEPSCs, and there was only a trend that was non-significant in synaptic strength, suggesting that mice and humans, maybe not surprisingly, are different, but for us surprising at this very basic level, and indicating that humans are more susceptible, more sensitive to heterozygous deletions of this particular gene. So heterozygous human but not mouse Neuroxin 1 mutant neurons exhibit a robust synaptic impairment that could serve as the basis for mechanistic and translational studies. Let me tell you about a second project, completely different, Synapsin 1. In fact, Synapsin was one of the first proteins I cloned when I started my lab. One of the first synaptic vesicle proteins that was ever molecularly investigated 30 years ago. In between, then others found that there are mutations in synapsin 1, an X-linked gene that predispose to autism and that are shown here. Now, synapsin is a multi-domain protein with a central region called C here that is an ATP binding region, and an N-terminal A domain that is subject to pKa-mediated regulation, phosphorylation, and that binds to membranes. We again generated conditional knockout neurons from humans. And in these knockout neurons, we completely deleted synapsin 1, as you see here, as a function of recombinase. As you can see here on the right, by this immunocytochemistry for synapsin, it's gone, as it should be. But the neurons are otherwise normal in terms of the synapse numbers, in terms of their morphology. So we now have a model system to investigate in a genetically controlled manner the effects of a synapsin mutation. And so we tested the functional effects again using a variety of approaches, including electrophysiology. And we did this in conjunction with phosphorylation that activates cyclic AMP synthesis because synapsin is such a prominent phosphoprotein. And what we found, as you can see here on the left, is that we compared the synapsin deleted versus the normal control neurons. We found a profound change, not initially, very different from the Neuroexistence mutation. Initially, the synaptic strength in the first response was normal. But during repeated responses, the strength of the synaptic response was decreased whenever synapsin was deleted. And this is illustrated in the cumulative trace at the bottom graph. When we looked at phosphorylation, we found that phosphorylation created the same phenotype in wild-type neurons as the synapsin knockout, and that the synapsin knockout prevented any effect of phosphorylation, as you see here on the right. So what these results show is that synapsin 1 deletion and phosphorylation treatment induce synaptic depression. They have the same phenotype, and that if you do one, you basically occlude the effect of the other, suggesting that in some ways, phosphorylation acts by inhibiting synapsin 1. So how does this work? And please recall, as I told you, that there's this N-terminal phosphorylation site in synapsin that mediates in this domain that mediates the binding of synapsin to membranes. And these are actually brings me back to studies that I performed in my lab 20 years ago, where we studied this biochemically. And what we found is shown in this very old graph from this paper in 1999 is shown here. On the left, you see synapsin A phosphorylation. On the right, you see the membrane binding. And when you look at the synapsin A phosphorylation, it is phosphorylated by cyclic AMP, shown here on the left, dependent protein kinase. And this phosphorylation is inhibited with a pKa inhibitor. When you look at the right at the same conditions, the phosphorylation-dependent protein kinase, the pKa-dependent phosphorylation of synapsin decreases its membrane binding. It increases its dissociation from vesicles. What you see here in this graph is the unbinding of synapsin. And if you block phosphorylation with an inhibitor, it blocks the effect. So this phosphorylation basically regulates the association of synapsin with synaptic vesicles, and in conjunction with the previous data suggests that pKa-induced synapsin 1 phosphorylation causes a synapsin 1 dissociation from vesicles that then has some effect that we saw in the electrophysiology. What is this effect? To test what this effect is, we looked at the synapse structure using fast freeze electron microscopy. And in these experiments that were carried out with Christian Rosenmund in Berlin, Germany, what we found, as shown here, is that the human synapses look like mouse synapses, beautiful, with lots of synaptic vesicles. And when we quantified the synaptic vesicles as a function of the synapsin deletion, we observed that there was a loss of synaptic vesicles by the synapsin deletion, as you see here in two and this was independent of stimulation. Stimulation itself caused a small decrease that was not significant, but even after 30 action potentials, there was synapsin deletion still caused a loss. When we looked at phosphorylation, as shown here at the right, phosphorylation alone also caused the loss of synaptic vesicles. And this is only 30 minutes of phosphorylation. So this is not days or anything. This is short-term phosphorylation. And this loss of synaptic vesicles was not associated with a loss of protein. So the vesicles are not destroyed. Instead, they are redistributed away from synapses. And the addition of the deletion of synapsin on top of phosphorylation caused a small but significant decrease, suggesting that phosphorylation alone didn't have the complete effect as does the synapsin deletion. So this shows that synapsin deletion and phosphorylation decrease vesicle numbers. And it also shows that the effect of synapsin gets less, although it's not completely abolished. There's no general loss of synapses. There's this loss of synaptic vesicles. There's no loss of synaptic vesicle protein. Now, can we visualize this loss of synaptic vesicles also in another way? And we can. We can do it by simple immunocytochemistry, by looking at immunocytochemistry for synaptic vesicle proteins in puncta, in synaptic puncta, which show a clear-cut phenotype of either phosphorylation or pre-dependent deletion of synapsin, as shown here for synaptotagmin or synaptophysin. When we compare that with a synapse marker, there's no change because the number of synapses are unchanged. It's the synaptic vesicles that are changed. And so the question then arises, having discovered a pathway here whereby PKA-induced synapsin phosphorylation leads to synapsin dissociation from vesicles and dispersion of synaptic vesicles, is this pathway controlled by a physiological signal, not just by phosphorylation, which is a pretty bad drug? You don't want to have it, right? Is this actually a physiological reaction? And so to address this, we turn to presynaptic neuromodulators, which are incredibly important in the brain. And we looked whether they would physiologically regulate synapsin phosphorylation. And what you see here is synapsin phosphorylation in human neurons. I'm not going to go through the detail. I just want to mention that serotonin increases phosphorylation and norepinephrine decreases phosphorylation. So physiologically acting neuromodulators can regulate synapsin phosphorylation in human neurons, and we can mimic the same thing with agonists for the receptors of these neuromodulators, and specifically with norepinephrine receptor acting agonists, as shown here on the right. And this then gives us a tool to check whether these neuromodulators will physiologically actually control synapsin phosphorylation dependent vesicle numbers. Okay, and to do this, we looked at this again. Now you see here an analysis of the synaptic vesicles using immunocytochemistry by measuring synaptophysin density and synaptotagmin density. And you can see that when you decrease the phosphorylation of synapsin with these agents, you actually increase the puncta number, suggesting, yes, you do have the same regulation by neuromodulators. When you knock out synapsin, as shown here in yellow, nothing happens because you basically it occludes the effect, right? You see the same thing for synapsin puncta, except that there's no synapsin left after the deletion. There's no effect on synapse numbers as measured with bassoon immunocytochemistry, as shown here on the right. So this, moreover, rescue experiments demonstrated that this is dependent on wild-type synapsin 1, and the phosphorylation site mutants of synapsin 1 also have the same phenotype, demonstrating that this is indeed the synapsin phosphorylation induced by neuromodulators that make this happen. So as a summary then, what I've told you here is, as a conclusion, that activation of presynaptic neuromodulator receptors, norepinephrine, serotonin, when they increase cyclic AMP, you cause a decrease in neurotransmitter release during sustained stimulation because you increase synapsin phosphorylation, which then causes synaptic vesicles to disperse from synaptic clusters. When you decrease cyclic AMP, you cause an increase in neurotransmitter release during sustained stimulation because of the opposite effect, because of a decrease in synapsin phosphorylation, causing an increase in synaptic vesicle numbers. Thus, this is a universal mechanism that regulates in a novel way how synapses function. Now, in the last minute or the last words of this presentation, I want to tell you that this is actually a universal mechanism that also applies to endocannabinoids, which are probably the most important neuromodulators in brain. They're the most important neuromodulators because they have the highest number of receptors and because they are also part of the response that we have, for example, for cannabinoids such as marijuana and their derivative drugs. And they are exactly subject to the same type of modulation. So what I've told you today in this very short presentation, which was pretty data rich, so I hope it wasn't too fast, it didn't just fast by. What I've told you today is that in 2 different projects, we used human neurons to transdifferentiate it from stem cells, iPS cells and ES cells, as a way to analyze the fundamental effects of disease-associated mutations. I've told you about Neuroxin 1 mutations and Synapsin 1 mutations. They differ dramatically in that the Synapsin 1 mutations always lead to disease, and whereas the Neuroxin 1 mutations lead to different types of disease that can be of different severity. They also differ in that the type of synaptic impairment that is created by these mutations is very different. In the one case, we have an effect that results in a loss of synaptic strength initially in response to a stimulus train. In the other case, synapsin, we have a loss of synaptic strength that accumulates during stimulus trains. I've also told you that these different mutations operate in completely different contexts. One is the synaptic vesicle protein synapsin that regulates synaptic vesicle availability for synaptic transmission. The other one is a synaptic adhesion molecule that regulates the strength of synapses in a manner that we incompletely understand at present. What these things, however, share is that these types of experiments enable us to gain a molecular, mechanistic insight into what these molecules actually do. And by gaining that insight, we achieve the first step, and this is only the first step, towards building an understanding of these diseases and identifying potential drug targets that might help us to actually treat these diseases. Because as it happens in the current neuroscience arena, in the disorders of the brain, the only diseases where there are reasonable treatments, as far as I know, are stroke and MS. And in these diseases, in stroke, this treatment that works is neurosurgical. In MS, MS is really a disease of the immunology system, of the immune system, and these are immunologically acting drugs. So we really don't have anything at this point in brain disorders that would help us treat these millions of patients who are suffering from these various different diseases. Okay, I finished my presentation and I think I'm in time, and I would like to I'd be happy to take any questions, but before I do that, I would like to acknowledge the people who performed the work I discussed. Chung-Hee Pak is a former postdoc who is now an assistant professor at the University of Massachusetts in Amherst. Tamas Danko is a research associate in Marius Wernick's lab Ingsha Zhang is a research associate in my lab, and Christopher Patzke, who did all the work on the synapsin studies, is now an assistant professor at Notre Dame University in Indiana. And this work was carried out in close collaboration with Marius Wernick and his laboratory, and also in part with Christian Rosenmund's laboratory in Berlin. We wouldn't be able to operate without funding, and the funding for our work that I discussed today is primarily from NIMH, with significant support by the Howard Hughes Medical Institute. I'd be happy to take any questions. Thank you very much.
Thank you very much, Professor Südhof, for your very, very exciting talk. So we have a lot of questions coming in. Before I start addressing or asking some of these questions, I have one of my own regarding your first project on Neuroxin 1. So you've shown that the Neuroxin 1 heterozygous deletions in the human induced neurons show this reduced synaptic strength. I was wondering if this is specific to the excitatory neurons, or would you also see that in the inhibitory neurons that you make with another method?
That's a great question, and I wish I could answer it. It's a question that we have obviously asked ourselves, but it hasn't been on the highest priority in addressing this. And obviously it's an important question. I don't know the answer to that question. It's a good question. I would suspect you would see the same impairment because it's a presynaptic impairment, and Neuroxin is present in both inhibitory and excitatory synapses. But I don't know the answer to that question. I'm sorry.
Sure, thank you. There's another question on Neuroxin 1 regarding this non-conservation between mice and humans. And do you think this means that humans are predisposed to neuropsychiatric disorders during evolution?
Well, the human brain arguably is more complex than those of most animals, although that sounds like a pretty anthropocentric view. Maybe that's not true. I don't know. So I think, at least in terms of rodents, the human brain is much more, or human neurons are much more susceptible to the mutation. Obviously, evolutionarily, this wouldn't show up because people with this mutation would presumably be evolutionarily disadvantaged, but the mutation is so rare, so you'd never see it. It's not the only case. It's been shown for other genes as well that humans are sometimes more susceptible, more sensitive to mutations. Yeah.
This leads me to a related question. It's more general, relating to model systems for modeling psychiatric disorders. So you've shown that mice aren't exactly great models because they don't really model human diseases. But on the other hand, what do you think about the use of human iPS cells as models for the disease, given that we can't see any behavioral defects in these neurons in culture?
Yes, I think that is another excellent question that we grapple with a lot, because it's not only for neuropsychiatric, it's also for neurodegenerative disorders that we have the same problem. I do believe we need to find the best compromise between the two principal systems, which are mice and humans. And I think we need to acknowledge at every step the limitations of these models. I think in particular that mice are sometimes, for human brain disorders, not only for human brain disorders, as a matter of fact, overinterpreted. I think it's valuable to do the studies in mice, and we do the studies in mice as well. I think it's important. I think for basic mechanisms, they are very, very, very useful. But I also think that some of these studies, major studies that are published, are not quite, maybe not quite appropriate in claiming that what is seen in mice is really that relevant. And, you know, I mean, all these studies about higher functions in mice that populate our journals, you know, about empathy and love and, you know, I think that that's all fantasy, that that's just, it's entertainment. It's basically, you know, like James Bond has very little relationship to reality. And you just have to be content at present that we can't really study these things, and doing these types of experiments and claiming these things in mice, to me, is, well, it's fun, but it's not exactly science. Yeah.
Sure. Moving on to some questions from the audience. So there was a question about a microenvironment around neurons and synapses, and is there such a thing that may play a role in neurodegenerative disease onset, in your opinion?
Oh yes, my God, yeah, especially neurodegenerative diseases. So I showed you that schizophrenia, GYSS, in the beginning of my talk, landscape, right? Most of these genes that you see there, almost all of them are neuronal. When you do the same thing for Alzheimer's disease, a large proportion are genes that are expressed in astrocytes or microglia. So there's a tremendous effect of the microenvironment and macroenvironment of neurons. In fact, the non-neuronal cells provide a major contribution to the function of the brain, for sure, to the function of neurons, and to the diseases of the brain. I think there's no doubt. And a lot of us are studying those contributions because we feel that we need to understand them in order to be able to gain insight into these diseases.
Thank you. Another question from the audience regarding endocannabinoids, which you've spoken about in your second talk. So is it possible to become schizophrenic after cannabis use, and could there be any mechanisms to trigger this?
I doubt it. I think my understanding is that cannabinoids are neither addictive nor are they inducing altered states that predisposes to psychosis. Very, very different from real hard drugs, from amphetamines or LSD or mind-altering drugs, you know, where I think there is a tremendous danger even. I think cannabinoids have danger for sure in the lives of individuals because of habit formation and altered mental states. But I'm actually almost convinced that these dangers are probably less than the dangers, let's say, of nicotine or of alcohol. Okay, so cannabinoids, the whole endocannabinoid system is incredible. It's undervalued, underunderstood because it's so diverse. Getting at this system with more specific manipulations will be one large opportunity, I believe, for in the future, certainly for drug development.
Sure. And regarding drug development on the cannabinoid receptors, this will be my last question from the audience. Do you think that this targeting of the cannabinoid receptors in the brain could represent a novel therapeutic target for any kind of neuropsychiatric conditions?
I believe that the receptors themselves are probably not the best target because there's only 2 receptors, and virtually all of the receptors that are important for the brain itself, virtually all of them are a receptor called CB1. It's a single receptor, and it's the same everywhere. So there are plenty of receptor agonists and antagonists. There have been some in the clinic. Some have failed miserably. So that is probably not the way to go. The way to go is to look at the machinery that regulates the release and synthesis and degradation of endocannabinoids, of the endogenous agonists of these compounds. And that's really where I believe are great opportunities.
Thank you for the insights, and thank you for the wonderful talk.
Thank you for having me. I hope that at least some of the audience got something out of it. And I appreciate everybody's listening to my talk. Thank you.